All right, well, this project got started when I heard Steve Orloff speaking about the damage he had been seeing and researching, particularly increasing frost damage in Northern California. I was also influenced by an article in Hay and Forage Grower where Mike Rankin interviewed a grower in Idaho who said their #1 issue was having more frost-resistant alfalfa. Now, in the case of Steve's plots, what we found was a lot of the damage, in addition to frost damage, was due to a disease that basically nobody had ever heard about, bacterial stem blight of alfalfa. Unfortunately, Steve passed away this last October, soon after we learned about our funding for this project. So we're working with NIFA to have Rob Wilson take his place as a co-PI. This is not my slide. Oh, okay, there I am. Woo! Everybody's nightmare. So the objectives of this research are to obtain information about the extent of this disease occurring in commercial alfalfa production fields, its association of the disease with frost damage, and the relationship of disease with bacterial populations. We want to identify DNA markers and candidate genes associated with resistance, and also develop germplasm with enhanced resistance to this disease, with the ultimate goal of delivering germplasm that has better disease resistance and hopefully better frost resistance for producers. So that you all will know more about this disease, here are some symptoms of the disease. It is always associated with frost damage, and it can reduce the amount of the first harvest by at least 50%. So we're hoping that increasing disease resistance will also reduce frost damage. The pathogen is called Pseudomonas syringae, Pathovar syringae, and here are the symptoms. You first get water soaking on the leaves. Then there also can be lesions on the stem that are first water soaked and then become brown with bacterial exudate. In the field, you see a lot of yellowing of the foliage and stunting. As the pathogen becomes systemic, it also starts a wilting phase of the symptoms, and you can have complete wilting and stunting of stems. So on the pathogen side, what we wanted to do is develop a sort of a fact sheet on bacterial stem blight. We'll be using a survey to query growers in California, Utah, Montana, Colorado, and North Dakota about this pathogen, whether they've seen the disease and how much damage it's causing. We'll be following the pathogen in new seedings over several years. And we currently have a very large collection from 2 years of research in 2016 and 17 that we're looking now at the genetic and pathogenic diversity in this bacterium. And one of the things we've done is we put a green fluorescent protein into the bacterium so that we can follow it and track it through the plant and understand more about how it's causing disease. On the host side, we've identified highly resistant plants, primarily from Germplasm and Maverick and ZG9830. We've produced F1 mapping populations by crossing individual resistant and susceptible plants. And then, oh, I'm sorry, time's up. A new student will be phenotyping these plants and developing resistant material.